So I'm a biologist and developmental biologist and energeticist, and most of my work over the last 25 years has been mainly about energy and its transformation. But thanks to some wonderful people, including Dr. Paolo Criticality, Maurizio Benfato, and Elizabeth Pace from INFN here actually in Frascati, I have begun to embrace information and entropy transformation as well, specifically in developing organisms. So this is a conceptual paper. I'm tossing out some new ideas, but it's based on about 25 years of data that I've been collecting. I was actually trained as an oceanographer originally. And as most oceanographers, when we go to the ocean, we always think about the beginning of life. So let's start at the beginning. And hydrothermal vents, which are features of this is the Mid-Atlantic Ridge. Right here, it runs down to the spine of the ocean. New oceanic crust forms at the Mid-Atlantic Ridge. In about 2000, there was a whole bunch of new hydrothermal vents discovered, actually, and it was called the Lost Cities because so much was discovered. But they were here at the very early history of the planet, and now we believe that this is where life on Earth first started. And so this has got back to the origins of life. And here at 6,200 feet, there are tremendous gradients, not only of temperature, but also of pressure and hypoxia as well. These organisms that you see here, these white organisms are shrimp, and they are entirely dependent upon chemical pathways, not light or oxygen for their metabolism. So there was a deep sea vent hypothesis originating from the fact that these extreme gradients, so I study energy flow down gradients. And so the mixture of hot water and the cold seawater created gradients suitable for the formation of organic molecules and I think other complex life forms, particularly the Riftia worms here, which are a remarkable complex group of organisms. And so this gave me the idea that stressors are chisels of evolution like Michelangelo and David. This is after a paper that was published in 2009 by DeBar, who's an immunologist. And I believe that stressors are important in generating life's complexity. They increase the rate of exchange between the organism and their environment, and those exchanges occur across gradients. And they could have originated from the deep sea vents, but also they are replicated in developing organisms. And I'm going to talk about this graph in detail in a minute. So changes in complexity patterns include changes in crucial and non-crucial events. And as we've heard a lot about this, I'll just reiterate that crucial events are generated by criticality, and namely, and this is where I want to emphasize, they are processes of phase or state transition from disorder to correlated disorder, affecting key organismal network functions. And as Bruce West and Paolo's have said in the book, Crucial Events, a subtle connection between informational exchange within and between networks and the complexity or non-simplicity of those networks. So I'm really interested in the corresponding informational change of entropy, as well as the biochemical change that causes energy to form along these gradients. Okay, so I think perhaps stressors, and this is my hypothesis, and criticality are chisels of evolution, particularly crucial events. Stressors such as high temperature, pressure, low oxygen, pH, all contribute to shifts in complexity and are considered to be, maybe along with criticality, chisels of evolution. Stressors particularly increase the exchange along gradients in and around non-thermal thermodynamic systems, which all living organisms are thermodynamic systems. Are non-thermodynamic systems. All living systems are open systems. So we go from the formation of the vents back about 4 billion years ago to when complex life first formed. About 3.5 billion years ago. This is the tree of life that you're seeing here. And all signaling transduction mechanisms that are conserved across all organisms, from bacteria all the way up to us, are the same. They are conserved among all living organisms. And they were established first about 3.5 billion years ago, thankfully, because they are very complex. And so we actually have to learn a set of them in order to understand how life works, at least at the biochemical level. So most of those gradients result in differentiation of our cells. So here is a morphogen. A morphogen is simply a protein that is secreted by a source cell. And the further you go from that morphogen, through diffusion, the less concentration of that morphogen. So you get a nice curve here. It is my suggestion that, well, we know that at certain concentrations of the morphogen, they trigger gene expression through epigenetic mechanisms. But what this is actually a modified graph from a paper that I published in Entropy in 2021. At the same time, so you have increased order coming and increased differentiation occurring at the same time as decreased order according to the information quantity or entropy flow. So energy and entropy flow are opposite to each other. And so what is interesting here is nobody's actually ever suggested this before. We don't have, I mean, it's reasonable in physics to expect this, but it would be really interesting to find out whether these points, these threshold points of concentration, are also perhaps crucial event derived. That's sort of what I'm guessing at. But these biochemical gradients that occur through the morphogen diffusion actually are responsible for the differentiation from a single cell to a complex cell. They're responsible for your fingers and your toes and your organs. That's how differentiation occurs, through specific gene expression as a function of these morphogens. So Schrödinger asked, how does a living organism avoid decay? That is, according to classical thermodynamics. Well, the answer is metabolism, from the Greek metabol, a change or exchange. Organisms stay alive by continually drawing from the environment, and he used the term negentropy, which isn't actually a correct term. It's really free energy. That is the quantity of energy available for organisms to actually do work, and it's directly proportional to the gradients organisms can use to maintain and reproduce. So we've got this common gradient theme here. Metabolism has become very sexy in the last 20 years because about 20 papers have been published by G.B. West, not Bruce West, and Galuli in Nature and PNAS. And they have used a fractal dynamics theory to explain this power relationship between metabolic rate and log mass. Here's the power relationship. And they have created what they call a universal three-quarter law. It doesn't have actually normal physical reasoning, but actually this slope extends all the way from unicellular organisms all the way to the Earth biomass. And so they said this is a universal law, a three-quarter law. Well, I was really interested in that because I studied developing organisms, and I quite frankly didn't believe it. So I tested it. I used a zebrafish, a developing organism here, and said, what do stressors do to this slope, to this scaling slope? We raised zebrafish under chronic temperature and hypoxia stressor conditions. And what you see here are, this is hypoxia, this is normoxia and normal temperature. This is high temperature and hypoxia, and this is high temperature normoxia. And in all cases, the slope was not equal to 0.75 significantly, but they were all significant. So here we have an extreme stressor, chronic stressor condition that changes the slope specifically in a developing organism. This is published in the Journal of Experimental Biology in 2022. All right, so what do you do when you add stressors? So you've raised these fish under chronic stress. What happens if you add even more stress? What happens to the system? Well, here's the one I just showed you, and these are acute. So this is even further acute exposure to temperature and hypoxia. And the tables in the paper are much more complete. But you can see the slope just basically becomes non-significant except for 2 points in which that could be simply measurement error. So what does that mean? Well, let's look at biology from a biological perspective, and then we'll get to the physics. Metabolic scaling relationships collapse, become non-significant when exposed to multiple stressors. Developing fish are unable to cope with long-term stress because of cumulative high energetic costs, which are referred to as allostatic loads, but can to some extent adjust to acute or short-term stressors. And I agree with Bruce. This is Bruce West, who spoke yesterday, and Damien West, his son, who published in 2013 a paper that said we need a novel nonlinear approach which may be suited to quantify how complex adaptive systems such as developing stages may respond to stage-specific selective pressures at the same time as coping with rapid self-organizational, developmental, and functional change, which is the characteristic of all developing organisms. So then I went to Ilya Prigogine, who stated that order emerges not in spite of chaos, but because of it. And his sentence here is really interesting. The greater the complexity of the system, the greater the amount of entropy it can dissipate. So all living systems are open systems. They take in energy and matter from the environment and dissipate the resulting entropy from the system into the environment. So what does this mean for, this is a series of cartoons, which actually reflect the empirical data that I just showed you on the scaling. So here is our control situation, our happy little larvae in the center here. Energy input equals energy dissipation. Everything is cool. Then you expose it to an acute stressor, and energy input is approximating energy dissipation, but the system still remains stable under acute short-term stress. What happens if you chronically raise embryos and further expose them to acute stress? That last experiment I showed you, well, you get entropic overload. Energy input is much, much greater than energy dissipation. The result, the system reaches maximum chaos, breakdown intensifies, and metabolic scaling relationships collapse. This is hypothetical. This is what I'm proposing. Okay. So most would say that if you have a complex adaptive system which breaks down, well, you have death. You know, it comes to equilibrium, it's dead. All living organisms don't want to be in equilibrium because they're dead. Well, I have an alternative suggestion. It's possible that complex adaptive systems may jump to a higher, maybe more resilient level with the capacity to increase dissipation. There's a new state evolved, perhaps as a function of crucial events. So these are new states possible. Sorry about the little fish there. But basically, this is a new state that's more resilient to chronic stress. This is really important now in our climatically challenged world. So let's have a brief, this is very basic. Developing organisms are complex adaptive systems that are exposed to multiple stressors may experience energy inputs from the environment that exceed their rates of dissipation. Thermodynamic gradients break down, resulting in changes in metabolic scaling relationships, but may allow for a new, more resilient, resulting in non-significant metabolic scaling relationships, but may allow for a new, more resilient state to ultimately evolve. This is hypothetical. So I created what is called, I propose a stress entropic model. There's a lot going on in this slide. I don't expect you to get it. I'll just show you some points. This was actually recently accepted with minor revisions in the Journal of Integrative Comparative Biology, actually 2 days ago, so I'm really pretty pleased. This will be published this year. But what I want to show you is these are the pictures I just showed you. Here is that new state. But stress response is intimately tied to immune development, immune system development. In fact, if you do not incur an acute stressor during development, you do not develop your immune system. So in this case, there's a really close correspondence between stress to a certain extent and a change of state and a development of your systems. So complex systems contain mixtures of patterns of complexity, non-crucial and crucial events. Crucial events are generated by criticality and the efficiency, and they are critical for the efficiency of information exchange. Crucial events indicate a change of state, which I think is going on here, as well as an immune response. Stressors, the question I have is, do stressors increase rates of state change? That's the question I'm going to be investigating for the next few years. How? By using biophotons. Actually, as measurements, since we've talked about biophotons, I won't define them. Obviously, thank you very much, Paolo and April, my student. And this is Maurizio Benfatto's result, and April talked a little bit about it. But she also, there's one thing that I wanted to address. In this seed that hasn't created the taproot, there is a very, very rapid DNA replication. And there was a paper back in 1966 by some Russians that showed that biophoton intensity is directly correlated to levels of DNA replication within the DNA, within the nucleus. But biophotons decrease the moment cell division, which is characteristic of taproot emergence, actually evolves. So one of the things that I want to do, and I hope April will work with me on this, is to test whether, if crucial events indicate an active change of state, here we have a change of morphological state and developmental state, will stressors increase the rate of state change? I don't know. There's some really possibilities here. So we hope to use stressors as an indication or as a test for this idea. All right, now I did say hemoglobin in the title. I've been studying hemoglobin for 20 years as well, and it's likely it's a complex molecule, it's an oxygen transporter. And the gene for hemoglobin, actually the ancestral gene evolved about 3.5 million years ago at the same time as complex systems did. And their origin might have taken place in the deep ocean rifts. I don't know, it's possible. So there's a lot of work done on hemoglobin. Some of the work I've done is on sickle cell anemia. And this is a disease that is caused by one gene mutation, actually one amino acid replication, a glutamine versus a valine, and it causes hemoglobin to come out of solution to form crystals in a sickle cell patient. And crystals that jam up into your capillaries are very painful, and many sickle cell patients die in their early 20s because it is an incredibly metabolically difficult disease. And during this state, it doesn't transport oxygen, so you get actually localized hypoxia in your joints particularly. So HBS, which is the mutation here, forms polymers, and you can see this is a blood cell. And you can see these polymers sticking out of the blood cell. And this is actually a burst blood cell, and you can imagine these are like needles in your joints if you're a sickle cell anemic patient. It's extremely painful. Well, it was hypothesized that it only occurs in humans, except I found it in fishes. About 13 species of fishes. I measured about 70 plus. This is published also in 2019 in this paper here. And what is really interesting is this ability for hemoglobin to crystallize is directly responsive to pH and hypoxia changes, oxygen changes. And it shows a change of state here and a change, here's the blood cells, but also the change in the pH. There is also a possibility that there's some crucial event occurring here as that goes from a solution to a crystalline state. So that's a guess, but again, this is something that we can test with biophotons as a measurement of changes of complexity, patterns of complexity. Okay, so this is my last slide. And again, this is just tossing out some ideas. But stressors and crucial events, are they both possible chisels of evolution? If crucial events are related to changes of state in the broadest sense, including causing dis-ease or a loss of complexity, thank you, Bruce West, and stressors increase rates of change, that is down gradients, then are crucial events like stressors chisels of evolution? Maybe not only in all living organisms undergoing change, but may have originated in complex molecules like hemoglobin at the beginning of life. The future work is, we'll use biophotons and statistical analysis to test with Elizabeth Pace and Maurizio how stresses are related to the rates of change of complexity patterns, i.e. crucial versus non-crucial events in organisms from beings, developing fish, and maybe even in complex molecules. Thank you very, very much.
Thank you. Thank you.